Good morning, children. This is Aniruddha, Mount Litra Z School, Jhansi. In the previous session, we have already discussed about cell cycle and cell division, and today we are going to start with photosynthesis in higher plants. Photosynthesis is an enzyme-regulated anabolic process of manufacturing of organic compounds inside the chlorophyll, containing cells from carbon dioxide and water with the help of sunlight as a source of energy. Photosynthesis is a chemical process uses light energy to synthesize organic compounds. That means light energy of sun has been converted into chemical energy, and that goes inside our body and that gives energy. Importance of photosynthesis. Photosynthesis gives us food. It is the primary source of food material. It releases O2, oxygen molecules, in the atmosphere. We all know that the plants are producers of any ecosystem, and plants produce food with the help of photosynthesis. And photosynthesis is done in the presence of carbon dioxide, oxygen, and this process takes place inside the chlorophylls, and chlorophylls are specifically found only in plants. At the early experiments, Joseph Priestley in 1770 for the first time performed a series of experiments with the help of bell jar, candle and mouse. And this experiment is very famous. We all have seen the diagram so many times in the books. In this particular experiment, a bell jar covers a burning candle and a mouse. And this experiment shows that mouse as well as candle, they both somewhere consume the oxygen molecules, because if the candle is burning, mouse starts suffocating. And when the entire oxygen of that jar is consumed, candle burning stops and the mouse also dies. And this particular experiment gives an idea that if we keep one plant along with a burning candle and mouse, they both survive for a longer time. Then in house showed that sunlight is essential to the plant process, that somehow purifies the air, followed by burning candle or breathing animals. He performed experiment with an aquatic plant and showed that in bright sunlight, small bubbles were formed around the green parts, while in the dark they did not. Later, he identified these bubbles to be of oxygen. Hence, he showed that it is only the green plants that could release oxygen molecules in the environment. And the picture here is shown before you. And this plant is aquatic plant Hydrilla. Then after, Julius von Sachs in 1854 again proved that plants produce glucose while growing and store them as a starch. He explained that plants have green substances which is present within a cell in specialized bodies. Today we know them as chloroplasts. He found that green parts in plant is where glucose is made, and that the glucose is usually stored as starch. Von Neil did one experiment with purple and green bacteria and demonstrated photosynthesis in a light-dependent process. He explained hydrogen from H2O reduces carbon dioxide to carbohydrate. He also concluded that oxygen comes from water and not from CO2. Finally, the correct equation for photosynthesis was discovered, and that is there CO2 plus H2O equals to C6H12O6 H2O plus O2. This is actually the reaction of photosynthesis. When we discovered the isotope of oxygen, that means radioactive oxygen, Samuel Rubin and Martin Kamin directly demonstrated that the source of oxygen formed in photosynthesis is H2O. The key features of photosynthesis was first explained in 19th century, and that plants could use light energy to make carbohydrates from CO2 and water. The empirical equation representing the total process of photosynthesis for oxygen evolving organisms was then understood as, where CH2O represented a carbohydrate. That means it is going to make glucose one day after combining with one another. Site for photosynthesis. The photosynthesis process takes place inside the chloroplast. The thylakoids in chloroplast contain most of pigments required for capturing solar energy to initiate photosynthesis. The membrane system, that means grana, is responsible for trapping the light energy and for synthesis of ATP and NADPH. Biosynthesis phase, that means dark reaction, is carried out in stroma where CO2 is reduced to carbohydrate. Pigments involved in photosynthesis are chlorophyll A, B, xanthophyll and carotenoids. In the blue and red light, the photosynthesis is the highest. The absorption spectra of pigment chlorophyll A and chlorophyll B show that absorption is highest in blue and red light. The least is green. Wavelength that optimally absorbed and used by green plants as the source of energy are in blue and red regions. There is a technique which is used to separate photosynthetic pigments. This technique is known as chromatography, through which we can take out the different pigments of photosynthesis. The structure of chlorophyll. It consists of a porphyrin ring with magnesium at the center. That means the central atom is magnesium, and there are certain hydrocarbon chains also. So here you can see a porphyrin ring and hydrocarbon chain, and this is a chain of porphyrin. In between there is one magnesium present. This magnesium is center atom. Photosynthetic pigments such as chlorophyll A and B have different respective works that they do in photosynthesis. Chlorophyll A acts as primary pigment, participates directly in light dependent reaction, converts light energy into chemical energy, whereas chlorophyll B acts as accessory pigment, captures light and transfers it to chlorophyll A, then initiates the light dependent reactions. Till now we have talked about the process of photosynthesis, the introduction. There are certain experimental proofs also in photosynthesis and the pigments of photosynthesis. In the next session we will be discussing about some different facts of light reaction and dark reaction. Thank you very much. Have a nice day.